Well, hello again, and it's good to be back in the afternoon, later afternoon. And I'm going to be talking about cellular mechanisms of omega-3 fatty acids in cardiovascular diseases. I'm going to talk very briefly about the artery and the heart, and then I'm going to switch to some novel approaches towards the major problem in cardiovascular disease, which is stroke. And we'll be talking about that. So if we look at the omega-6 and the omega-3 fatty acids, we can see the omega-6 are defined as fatty acids in which the first double bond is 6 carbons from the methyl terminal, and the omega-3 are the first double bond is 3 carbons from the methyl terminal. Now, both these fatty acids, we need the essential fatty acids, linoleic acid for the omega-6 series, which we get largely from vegetable oils, and the omega-3 fatty acid, even fish. Fish have to have the essential fatty acid, alpha-linolenic acid, we get from very small shrimp and algae. And then both chains of fatty acids, the omega-6 and the omega-3, are elongated by the same enzymes to make them longer and desaturated. The omega-6 chain forms arachidonic acid, which in general, not totally, is pro-inflammatory, pro-apoptotic, and really does things that aren't so good. On the other hand, on the omega-3 side, we have elongation to EPA, eicosapentaenoic acid, which is very, very anti-inflammatory, anti-apoptotic, and does good things, beneficial. And that EPA gets elongated further to DHA, docosahexaenoic acid, which is a very important structural fatty acid. 50% of the fatty acids in the retina are DHA. Depending on the phospholipid subspecies in the brain, 15 to 22% of the fatty acids are DHA. But in addition, it's also very anti-inflammatory, as we'll see, and does a number of things to prevent cell death. Both the EPA and DHA produce other kinds of smaller molecules when they're catabolized, which are called resolvins and protectins, which are also very effective in protecting against inflammation, apoptosis, and a number of diseases, and we'll talk about them soon. So if we look at fatty acids and what do they do in the cell and cell biology, they are active in inflammatory responses, apoptosis, intracellular signaling pathways. And we've done quite a few studies. The yellow things here are pathways that my laboratory has focused on at Columbia, and as well the orange at the bottom, other areas. And we have found they're important in gene regulation, quite a few genes, antioxidants, and mitochondrial function. If we look at the proposed roles of omega-3 fatty acids in human health, they're listed all over the world. That infant formulas, which are the replacement for human breast milk when people are not breastfeeding, are now essentially all fortified with DHA. They have positive effect in pregnancy outcomes, perhaps some cancers, factors associated with metabolic syndrome, and they don't reverse neurological degeneration, but they actually may delay the progression of some of the neurological degeneration problems. And quite a number of studies in suicide prevention, depression, show effectiveness of omega-3 fatty acids, especially EPA. They're used to treat hypertriglyceridemia, and they also decrease liver triglyceride in some forms of non-alcoholic fatty liver disease, depending on the genotype of the subject that has non-alcoholic fatty liver disease. Today I'm going to really focus on things at the bottom of the slide, which is some focus on atherosclerosis and myocardial infarction, and then we're going to get into the brain and talk about stroke and a bit about traumatic brain injury. So this is a slide where we take mice that do not have LDL receptors, so they're LDL receptor knockout mice. And these mice, when we put them on a high-fat, saturated fat diet, Western diet, if you will, develop atherosclerosis very quickly with high cholesterol. And on the top part of the slide, so we feed them the chow diet, which is the control low-fat diet, or we feed them the high saturated fat diet, which is high in total fat, 42%, and in cholesterol. And we feed them a high-fat omega-3 diet, which is also high in cholesterol and high in total fat. And after 12 weeks, we sacrifice the mice. And this is what we're looking at is cross-sections of the aorta taken just above the innominate artery, just above the aortic valve, sorry. Okay, cross-sections of the aorta just across the aortic valve. And here you can see on the low-fat chow diet, this is the aortic valve. This is the arterial wall. And we're staining now with oil red O to stain for triglyceride and cholesterol esters, neutral fat lipid. And you can see with the saturated fat, we have diet, we have a lot of atherosclerosis, atherosclerotic lesions. That's in red here and thickening. But with the omega-3, we have very little. And we can actually quantitate the atherosclerotic lesions here by doing the mean lesion area of the different parts of the aorta. And you can see here is the chow fat diet, low fat. Huge increase in lesions with the saturated fat. And then we continue the high saturated fat diet, but replace, with increasing ratios, replace a bit of the saturated fat with omega-3 fats. And you can see that even with low amounts of omega-3, we totally wipe out the bad effects of the saturated fat, even though it's still a high-fat, high-cholesterol diet. And these are different ratios here at the bottom. So in the face of a Western diet, a saturated diet, with a bit of omega-3, we stop atherosclerosis progression. And I'm not going to show you, but we have quite a number of pathways which we now understand on the effects of omega-3 fatty acids in the arterial wall in decreasing atherosclerosis by decreasing arterial lipid deposition, decreasing pro-inflammatory cytokines, pro-inflammatory cells, and increasing anti-inflammatory mediators. And these are some of the mechanisms that we've published upon over the last few years. So what I've been talking about now has been dietary changes. And then there's most of the literature on omega-3 that's out, people study, is on long-term supplementation with omega-3 supplements or with high amounts of fish in the diet. We're now going to ask a novel question, which has not been studied so well. And Dr. Carpentier referred to it somewhat this morning. What about if you give acute administration of omega-3 after an event? Or you mentioned before and after cardiac surgery, but we'll now look at heart attacks and strokes acutely, where we give injections of omega-3 fatty acids after the event in what we call bolus injections. And the question is, will acute injection of omega-3 fatty acids packaged in, I'll show you, triglycerides, protect the heart and the brain after injury. And we're looking at ischemia reperfusion injury, which is sort of like what happens in heart attacks and what happens in strokes in humans. So this slide is a slide of heart attack in mouse. Okay? And we do this by tying off the left anterior descending artery, coronary artery, to the left ventricle for 30 minutes and then releasing it. Okay? So it's a 30-minute blockage, which causes a heart attack or a stroke, and a heart attack in mice. And then after that, we inject either normal saline, which is the control here shown in green, or we inject an omega-3 triglyceride emulsion in which 46 to 48% of the fatty acids are EPA and DHA. And then we sacrifice the mice at 48 hours. So here on the upper left, you see the heart. This is the left ventricle. And this is the infarct area in the saline injected mice. And that's the infarct area in the mice that have received the omega-3 emulsion. And that's quantitated here in terms of infarct size with the saline injection and huge decrease in infarct size with the omega-3 triglyceride injection. And below, lower right, you see decreased release of enzymes. We have more recent studies showing that the enzymes you measure in the ER, like troponin and CPK, are also markedly reduced after we inject omega-3. And on the lower right, you see echocardiograms and ECG, which show, I'm not going to go through the details, but we preserve myocardial infarction. Myocardial function, not infarction. We preserve myocardial function. And this is just one example of many mechanisms that we've studied here to understand why we protect the heart. And this just shows a factor called BCL2, which is anti-apoptotic, so against cell death. And you can see that these are normal control mice, no exposure. These are the ones that have had the heart attack and with saline treatment, and marked increases in this protective BCL2 after we inject the omega-3 triglyceride emulsion. So we hypothesize that acute injection of omega-3 in humans after MI will decrease morbidity and mortality because of reduced infarction size and recovery of cardiac function. And at the bottom of the slide, you see the summary, which we've actually published, where we show decreases in ischemic markers, decreased arrhythmias, decreased in autophagy and apoptosis, but we preserve myocardial function and myocyte survival. Now, let's go a little higher. We can go up to the brain. We're going to talk initially about ischemic stroke. And so myocardial infarction has a lot of good treatments, and it's getting better. There's really no great treatments out there for stroke. Very effective post-stroke treatments are not available, and ischemic strokes account for about 87% of the strokes in the United States and about 50% of the strokes in China. China, you also have hemorrhagic stroke as a major cause. But the interesting thing is, and we can maybe talk about this later, the causes of cell death in hemorrhagic stroke, the cellular mechanisms, are actually quite similar to ischemic stroke. And if we look at the, what's the pathobiology in ischemic brain, so this is not just brain, but other tissues will show some of these pathways. So you can see that we have many pathways that are involved after loss of oxygen or loss of blood flow. Some occur, within minutes, some within hours, and some within days. And in the green circle are pathways that our laboratory has studied in terms of mechanisms and understanding the pathobiology of brain damage. So we see that there's calcium and calcium buffering capacity, reactive oxygen species, mitochondria function, lipid peroxidation, actually protein 2, inflammatory markers, etc. So we actually hypothesize that these different pathways might be affected in a positive way by omega-3 fatty acids. And the hypothesis is true. So these are experiments in 3 rodent species. You have to look at me for a second now, here, where we tie off, put a ligature on the right carotid artery, which in a rodent, a rat or a mouse, is not enough to cause a stroke because they have a very good circle of Willis. So you have to put them in a hypoxic chamber. Lower the oxygen to 8% for about 15 minutes, and then you put them back into room air. And then when we put them back into room air, we inject a lipid emulsion containing EPA and DHA, or DHA and or EPA, I'll mention that soon, or normal saline. Okay, and then 24 hours later, we sacrifice the mice and do neurofunctional tests and histological and mitochondrial function tests. So you can see here on the top part of the slide, let's take the brain here to here. We do coronal sections from the front to the back to the brain. I'm not showing them all, but you can quantitate the areas of injury, the degree of injury, and the infarct size. And that's shown on the top with 2 slices, with 1 slice here. So this is TTC staining, and this is on the right side. The live brain is red, the dead brain infarct is white. We can quantitate it with NIHJ imagers, and below we show actually in red here the injury scores or infarct size of mice that have been injected with saline after the injury, or mice that have been injected, or rats with omega-3 in blue after the stroke injury. We've done this also in another model called right middle cerebral artery blockage, which is direct blockage, and we get exactly the same results. And so we see marked protection against infarct. Now what happens if we make emulsions which contain only DHA in the triglycerides or only EPA? Well, surprisingly, we found that only DHA is effective as compared to EPA, and that's shown here with, again, saline, EPA triglyceride, DHA triglyceride, and the bottom is the pictures. In the United States, until about a year ago, every patient who got lipid emulsions got intralipid, and now there's another one that's come out with fish oil and medium chain triglyceride, Dr. Carpentier talked about. But look what happens if we compare saline, omega-3 oil emulsions, or this is intralipid. So intralipid makes the infarct worse. So it's something to consider in hospitals. We don't know, we would still have to do the proper trials, but to me this is very worrisome, especially if you have patients with some kind of brain injury problem. Something to think about. And the other thing is, it's very important if we're going to have a therapy for stroke, we need what's called a window. You know, how long before the patient has the stroke that you can give the injection. So this slide shows, these are in rats with right middle carotid artery occlusion, that if we inject saline at 0 hours right after the stroke, or 6 hours is the damage. But now let's look at the window 0, 4, 6 hours with the omega-3 injection. You can see we have a window of up to 6 hours. There's nothing out there, TPA or the other kind of treatment for stroke, except for thrombectomy now actually, that can give this kind of protection. There's no agents that have a 6-hour window in terms of indication. And if we start to look at a mechanism, we looked at quite a few, I'm only going to show you a couple of slides I think, is if we look at mitochondria that we isolate from the brain, 30 to 60 minutes after the injury. That's before you see any histological changes of injury. And we isolate the mitochondria and look at the production of reactive oxygen species. This is very consistent in different models. Here's saline, and here's the omega-3 treated mice. Big decreases in reactive oxygen species. Just Dr. Ginsburg mentioned earlier that Ed Fisher found that the decrease in VLDL production was associated with lipid peroxidation. We find the opposite in the brain, one. And two, the other thing is the omega-3 fatty acids, that's why they're used in hypertriglyceridemia, actually decrease triglyceride production in the liver, and combined with decreased ApoB secretion, lower triglycerides. So this is just one of the mechanisms. So we can summarize, and I'm not showing you all the data, but we now have actually 6 rodent models. We decrease brain edema. We have increased levels of DHA, and I'll show you in a minute its catabolite, this neuroprotectin D1, which is protective 2. We decrease mitochondrial ROS and whole brain ROS production, and we preserve mitochondrial function as we measure by one measure, calcium buffering capacity. So all the data that I've shown you so far is after 24 hours. What happens if you do not give the mice any more omega-3 but wait 8 weeks later? And we find that 8 weeks later, after just the initial injection of the omega-3, we don't give any omega-3 in between, that we have much less brain shrinkage, so the brain stays big. But importantly, this is very important, we preserve neurofunction. We do neurofunctional tests, about 4 or 5 of them. So with the one injection 8 weeks before, we not only get decreased infarct size, but we get preservation of neurological function, especially with DHA, not so much with EPA. So just we'll take a quick look now. I mentioned these derivatives of arachidonate and DHA. So we'll look first at here's the DHA derivatives that come from the DHA in the plasma membrane. After ischemic injury, they get released. And then we produce these different resolvins, mucins, and we're interested particularly because we have found this to be most effective of all the catabolites, neuroprotectin D1, NPD1. We've also looked at arachidonic acid metabolites, catabolites. They're listed here, and they are indeed pro-inflammatory and don't do any good. So I'm just going to show you a couple of results for NPD1, which does good after stroke. And this shows that DHA increases in the brain after we give the DHA injections, and that's shown here with hydroxy DHA, and this is NPD1. So big increases after the administration of exogenous DHA on the neuroprotectin D1 levels. We've shown that NPD1 decreases post infarct volume, but not as much as DHA does generally. So that's interesting. And that's shown here on top, and then the pictures are below. We're also doing, and this is work of Hilda Zerporli, she's doing quite a bit of work on the different transcription factors, proteins that are important in mitochondrial death and mitochondrial non-death, saving mitochondria. These are called BACs and BAC and caspases, etc. I'm not going to show you any of the data, but just in general, we're getting very, very interesting results on how the omega-3 actually protect against pathways that kill mitochondria and kill cells. So that if we look at what NPD1 can do, they act directly on brain mitochondria. This is new. There's no papers describing mitochondrial function in these protectins. And these are the functions where NPD1 has cellular effects. which I'm not going to go into. And so we know that DHA promotes neuroprotection via multiple and interactive mechanisms, and the DHA and NPD1 share similar neuroprotective pathways. And we're trying to answer the question right now, does DHA have a wider range of molecular actions compared to neuroprotectant NPD1? And we think likely yes. So I think we can conclude that omega-3 glyceride emulsions could be new pleiotropic, multiple mechanism bioactive molecules to meet unmet needs for stroke and other organ injuries. We're very excited about the work. We think that the reason that so many other stroke agents that have been tried in animals haven't worked is they only target one or two mechanisms. The omega-3 target at least 9 mechanisms that we're aware of so far, most of them in mitochondria. And with that,